Hello guys and welcome back to Excelsior Academy. So today we are going to do the last cytoskeleton filaments, and that is intermediate filaments. Okay, so this is how, again, this is one example where they have mentioned about the keratin intermediate filament network. Here you can see the question is like, in that small cell along with all the actin filaments, microtubules and intermediate filament, the cell is totally crowded, so there is no space. Okay, still many organelles are still arranged very properly. Now, these intermediate filaments, they have got diameter of around 10 to 12 nanometer, which is like in between of actin filaments and the microtubules. They are not directly involved in the cell movements, but they play important role in the by providing the structural role, giving the mechanical strength to the cells, tissues, and provide a scaffold for the localization of cellular process, including intracellular signaling. So later, after 1 or 2 units, we'll see in the cell signaling that these intermediate filaments are involved in the cell signaling. Like many proteins are there, which is in direct connections with these intermediate filaments. दो cell को जोड़ने के लिए भी these intermediate filaments are playing very important role, and also like the cell along with the extracellular matrix. So that connection is also via the intermediate filaments. Now, while cytoplasmic intermediate filaments are characteristics of most multicellular animals, they are not found in yeast, plants, and some insects. Okay. So please remember that they are present in the animals, but they are not found in the yeast, plants, and some of the insects. Please remember this. Now, these are the different types of keratins. And yes, this table you have to remember. This is important. ये आपको याद करना ही पड़ेगा. Okay. So, type 1, type 2, 3, 4, and 5. Type 1 is your acidic keratins. Site of expression is epithelial cells. Neutral or basic keratins, epithelial cells. Okay, so question comes like whether the basic keratin comes under the type 1 or type 2. ऐसे question आएंगे. Okay, now coming to the third type, vimentin, desmin, glial fibrillary acidic protein, peripherin. Now, ये तो नाम से ही first of all you can remember like where all these are getting expressed. For example, peripherin is in the peripheral neurons. Glial means in the glial cells. Desmin, muscle cells. Vimentin, fibroblast, WBC, and other cell types. Okay. Now you need to remember all these things, because as we progress in this unit and also in the next unit where we will talk about the extracellular matrix proteins, and where we talked about the desmosins and hemidesmosomes. So there all these proteins are needed. Okay, so you have to by heart. Let me go slowly. So again, acidic and basic keratins. So keratins are present in your epithelial cells. Okay. Then comes the vimentin, desmin, glial, peripherin. Vimentin again in the fibroblast, desmin muscle, glial, peripheral neurons. Okay. Now coming to the neurofilament proteins. नाम से ही पता चल रहा है that it is expressed in the neurons. Okay. Now, NF, L, M, and H, light, medium, heavy. Okay. NF stands for neurofilament proteins. All are expressed in your neurons. Then alpha internexin, again, neurons. Nestin, stem cells. Nuclear lamins, ये तो मालूम ही होगा. Where is it? It is the nuclear lamina of all the cell types. Okay. So under this, in the intermediate filaments, you have got the nuclear lamins. Remember, we have talked about the IF filaments known as lamin A, B and C. So A and C है जो आपके एक साथ ऐसे helical structures बनते हैं, and they are arranged around the, below the nuclear envelope. And now these are connected with the help of the lamin B. Okay. Is this clear to everyone? याद हो गया? Should I ask? Okay, let me ask then. Akshay, where desmin is expressed? Tanvi, nestin?
muscle cell.
No, for you, I asked nestin. Desmin is expressed in the muscle cells. What about nestin? Okay. Krishita, alpha internexin?
Fibroblast.
Where?
Fibroblast.
No. Last, Shruti, Vimentin? किसी को नहीं आ रहा. Not paying attention, it means. See, Vimentin is fibroblast, desmin is muscle, internexin neurons, nestin stem cells. Okay, it's okay. It was just first time, but I might ask any time in the future classes. Okay, and this everything is present in your Cooper reference book. Okay, now assembly of intermediate filaments. Let me know, guys, if you don't have Cooper ka PDF. Okay, no need to buy. If you can study through PDF, then let me know. I can share with you. Okay, now coming back over here, assembly of intermediate filaments. Despite considerable diversity in size and amino acid sequence, intermediate filaments share a common structural organization. Okay, now that is, you have got the alpha helical. So they are all made of alpha helical rods. Okay, they have got the head and the tail. Always your head is present in the N terminal, and then the C terminal, you have got the tail. So they become, so two polypeptide chain come together, they form the coil structure, forms a dimer, and then again these two come together to form a tetramer. So this is how together all these are forming a protofilament, right? And then together it forms a filament. Now now at least 8 protofilaments are present in one 8 protofilaments are present in one filament. For example, if we see in the case of the microtubules, we know that around 13 protofilaments are required, right, to form one microtubule. In the same way, intermediate filaments, here you need at least 8. Okay. Now, this is also can be better understand when we talk about the lamins. Okay, we'll come to that. Now, filament assembly, this is the same thing which we just discussed in the table. For filament assembly requires interactions between specific types of intermediate filaments. Example, keratin filaments are always assembled from heterodimers containing one type 1 and one type 2 polypeptide. Okay, whereas the type 3 proteins can assemble into filaments containing only a single peptide, polypeptide, vimentin, or it will consist of two different type 3 proteins like vimentin plus desmin. Okay, now type 3 proteins do not however form copolymers with the keratin. So what we are saying here is that keratin may type 1 can interact with type 2 intermediate filaments. Okay, now in the case of type 3, it will interact only with another type of type 3, that is vimentin or desmin, या ये इन दोनों का combination होगा. But type 3 proteins will not interact with the type 1 and type 2. Okay, I hope you got the difference here. Intermediate filaments are generally more stable than actin filaments or microtubules and do not exhibit dynamic behaviors controlled by nucleotide binding that are associated with these other elements of the cytoskeleton. So what does it mean? What does it mean that they are more stable? It means that in contrast to microtubules and actin filaments, remember we have talked about the treadmilling over there. So what does this treadmilling means? It means at this plus and a minus end is there. So here keep on adding of actin, or we say tubulin. Okay. And here it keeps on depolymerizing. So this was the dynamic instability of actin as well as microtubules, right? But this is not seen in the case of intermediate filaments. So that's what we say, that they are more stable and do not exhibit the dynamic behaviors, which is controlled by nucleotide binding. If you remember, actin में हम ATP bind करते थे, tubulin में हम GTP bind करते थे. Okay. However, intermediate filaments are dynamic within the cell. For example, इनके regulations के लिए. So over there, it was nucleotide binding regulated. Here it is the chemical modification which results in the regulation of intermediate filaments. For example, phosphorylation, which will regulate its assembly and disassembly. Okay, guys, are you able to follow me? Akshay, able to follow?
Yes, ma'am.
Okay. So one example is phosphorylation of the nuclear lamins. Let's take this example of nuclear lamins. So results of disassembly of the nuclear lamina and breakdown of the nuclear envelope during the mitosis. So in short, what is happening is that when nuclear envelope disappears, as I said before also in one of the lectures, that it doesn't mean that koi magic ho raha hai. It is getting disappear means the envelope is broken down into small bits, right? And that is happening because the lamina is getting phosphorylated. So those lamin proteins A and C, they are getting actually phosphorylated. Okay, so what happens? The nuclear envelope disappears. They are broken down into small lamina. Okay, now once this gets dephosphorylation, again they come together and join to form a nuclear envelope. Okay, now cytoplasmic intermediate filaments such as vimentin, there are also phosphorylated, which can lead to their disassembly and reorganization in dividing or migrating cells. So here it says that this slide, okay, the takeaway message of this slide is that the phosphorylation is a process by which the intermediate filaments can be regulated for their assembly and disassembly. Okay, so this is how, again, the same thing, monomer, dimer, two types of intermediate filaments can come together, or the same family or the same type of intermediate filaments can come together, then they form a polymer, and then again protofilaments and the intermediate filament. Okay, now where is your lamina? Lamins over here. So this is your NPC. Just below the nuclear membrane, you have got the lamins present over here. Can you see this red color things? They have shown that this is your lamins, and below that you have got your chromatin, which is right now not condensed. Okay, now here, can anyone tell me which color is indicating the lamins? Krishita, would you like to try?
Maybe green.
Any other person?
That would be active.
Shruti? Okay, Tadvi, would you like to try which color? Okay, back to Akshaya.
Blue.
Which one?
Blue. Blue.
Okay. एक ने blue बोला, एक ने green बोला. Now only remaining one is the pink one, which is the correct answer. The pink color is the correct answer. See, why I ask this question, because already I have mentioned you that blue is always your DAPI, which is going to stain your nucleus. वो भी क्या? The AT rich region of your DNA. Okay, तो जहां जहां आपका chromatid में AT rich region है, that is going to get stained by the DAPI, and that will look like your blue color. Okay, now right now what just I explained to you, that this is your nucleus. यहाँ पे आपका NPC था, उसके below I showed you the lamins just now, correct? ऐसे lamins are present. Now what do we see here? You see that pink color का outer boundary है, उसके अंदर blue है. This blue is because of your DNA. And this pink color is because of lamins. उसके ऊपर कोई color नहीं है ना, because nuclear membrane is not being colored over here. Did you get the point? और green color क्या है आपका यहाँ पे? The other intermediate filaments. or actin or microtubules. It can be anything which is present outside the nucleus. Did you get it?
Yes, ma'am.
See, that's why I'm saying when we do all these experiments, the person who is doing, they should understand like where all these things are present. Now here recently one question was there. I don't know whether it was interview question or YouTube में मुझसे किसी ने पूछा था. I just don't remember like where this question was asked. So the question was asked like, how do you understand which protein is expressed in which organelle and where? कैसे पता चलेगा? So simple is that if you know about that protein, of course here they have used the antibody as your anti-C lamin or anti-lamin C protein antibody they have used, right? And they are being tagged with anything. So let it be any kind of antibody which is tagged to any color. The point is that blue is always going to be your DNA. ठीक है? By default it is always going to be the DNA because हमने अभी तक और कोई डाई नहीं बना पाया है. Mainly what color do you see is आपको red fluorescence मिलेगा. अभी ये पिंक इसलिए दिख रहा है because of the ये blue के वजह से पिंक आ रहा है, नहीं तो ये भी red ही है. And then green. Blue is for your DNA. So majority time you will get all these colors only. Now there are few cases depending on the pH you can also get orange. ठीक है, इससे ज्यादा कोई कलर वेरिएशंस आपको नहीं मिलेगा इन द फ्लोरेसेंस. वी आर रिस्ट्रिक्टेड टू दैट मच ओनली. ओके, नाउ व्हेन द क्वेश्चन वाज आस्क्ड कि ये क्या है, सो of course we are talking about the lamins, of course we are talking about the intermediate filaments, so that's how you should guess also कि ये उससे co-related होगा. Okay, चलो, everyone got the point over here, like why this pink color border is the answer. Those are the lamins, okay, which are surrounding your nucleus. Okay, now this figure is really very important. Listen carefully because I'm going to ask one of you to explain it again. Okay, so here you have got your nucleus. It is present in the interface. Okay, now you have got the NPC, the green color things are the lamins, and inside you have got the your chromatin, the DNA. Okay, now what is happening is that the phosphorylation of lamin protein is happening. So what will happen? This will result in the disappearance of your nuclear envelope because lamins are phosphorylated. Okay, so this green colored intermediate filaments are now phosphorylated. So now you can see they are single. Correct? यहाँ पे क्या है? They are coiled structures. But after phosphorylation, they become single and entire nuclear membrane has been disappeared. So ये ऐसे होगा, आपके छोटे fragments बन जाते हैं, ठीक है? When the lamins are phosphorylated, by that time your DNA is getting condensed into the chromosome. Okay, now suppose the separation has happened. एक chromosome वहां चला गया, एक chromosome हमारे पास आ गया. Okay, now what will happen? In the cell cycle, you will get this that dephosphorylation of lamins will start. So what does it mean? The moment these phosphate groups are removed, okay, the lamins will come together now. जो ऐसे single है, now they will be a coiled structure and they will start arranging themselves like this. This will make sure that this nuclear envelope, okay, they are nothing but what? They are phospholipids, right? So phospholipids will try to attach to each other. तो ये कैसे करते हैं? क्रोमोसोम के आजू बाजू में lamins आ जाते हैं, उसके आजू बाजू में your nuclear envelope will come. Then slowly when they form these kinds of structures, ऐसे. Okay, so this is only 2 chromatids they have shown. So we know that in each case we are going to have the 23 pairs are going to be there, right? So they come together and now these start fusing. This is phospholipid mate, this is also phospholipid mate, right? So they start coming fusing and finally they fuse together and you get back to the interface of the cell stage. Clear? Any doubts? Any step? Yes, Tanvi, is it clear?
Yes, ma'am.
Okay. Now, so intracellular organization of the intermediate filaments, like where exactly they are present is the question. So they form an elaborate network in the cytoplasm of most cells, extending from a ring surrounding the nucleus to the plasma membrane. So again, they are present in the cytoplasm. Okay. Now both keratin and vimentin filaments, they are attached to the nuclear envelope, apparently serving to position and anchor the nucleus within the cell. So if the question comes like, what makes your nucleus attached to only one position or one localization inside the cell? The answer is that the keratin and the vimentin filaments are making sure that the nucleus is anchored in one place inside the cell cytoplasm. Okay. So this is again the antibodies are used against the keratin filaments over here, and you can see that the entire place it is present. This is of the epithelial chain. Okay. So the keratin filaments of epithelial cells are tightly anchored to the plasma membrane at two areas of specialized cell contacts known as desmosomes and the hemidesmosomes. Okay, अब यहाँ से थोड़ा complicated होगा. So here also it is just the introduction. Okay, desmosome and hemidesmosomes. We will be doing this in detail in the upcoming lectures. Okay, now what is desmosomes? So desmosomes is between the two adjacent cells. Okay, so this is one and this is another and this is outside of the cell. I hope you're getting it right. So this is one particular cell like this. This is another cell. Now, what do we see in the desmosomes? The two cells are making connections. Like how? So desmoplakin. Okay, desmoplakin is having your both desmocolin and desmoglin. These are the two proteins which is making connections with the other cell car, desmoglin and desmocolin. Okay, and again this is desmoplakin which is in connection with connection with the desmocolin and glin with the help of the plakophilin proteins and plakoglobin proteins. Okay, these are your plakophilin, these are your plakoglobin, and all this are in connections with the intermediate filaments. Okay, did you get it? So first is the intermediate filaments. They are attached to your desmoplakin. They are attached to your plakoglobin plus plakophilin. It is written as PKP, plakophilin. Now these are then attached to your, they are attached to your desmoglin and desmocolin. Okay, ये एक side का हुआ है. Now desmoglin and desmocolin of another cell, again plakoglobin, plakophilin, then again you have got the desmoplakin and the intermediate filaments. Okay, so this is desmosomes. Now what is the function of desmosomes, why it is important, everything we'll cover in the upcoming lectures. In short, any mutation to this will result in cancer. For example, when I was working at Tata Cancer Hospital, I was working on this plakophilin. Okay, and plakophilin में भी बहुत तरह के types होते हैं. So I was working on PKP3. There are 1, 2, and many are there. So I was working on this, and what I found that if this protein is mutated, okay, it results in colorectal cancer. So though we feel that this is very small, but we see what happens is that any mutation to any of these proteins will result in cancer. Okay, is everything is clear? I'm gonna ask one person to explain now. Let me pick the name. Okay, I'll go with Krishita. Yes, Krishita, would you like to try explaining this, the desmosome connection?
Yes, so there is this intermediate filament within the cell, which is attached to the desmoplakin,
Correct.
And then desmoplakin is attached to the plakophilin and plakoglobin.
Correct.
So the purple one is plakoglobin, and the light purple one is plakophilin, pink more
Hmm.
of.
Hmm.
And then that is attached to the desmoglin and desmocolin,
Correct. Correct.
which is present outside of the cell, and they are connecting the two cells.
Yes. Yes.
Okay.
Good. So that's really good, Krishita. So one suggestion over here, whenever you get to study yourself, okay, try to repeat like how right now I asked Krishita to repeat, right? Then repeat, close your eyes and visualize a figure. And of course, when you study, focus on one particular type of figure, because see, for desmosomes in the internet, in the Google images, you can get many different types of images saying the same story, or maybe some additional proteins will be also mentioned, but you focus or you try to take only one figure, try to remember in that figure only, close your eyes and just imagine that that figure, and try to understand and remember the protein, then it really becomes easy. Okay, this is really my personal experience, I'm telling you. That's why for me few images are like very specific. अगर मैं पढ़ाते भी हूँ तो मुझे वही images चाहिए. I know in the Google I can get many, but I go with that figure only because that's how I have learned, and I remember now also, even though I cleared my exam in 2009 and 10. Okay, now coming to the hemidesmosomes. So hemidesmosome is between the cell and your extracellular matrix. Remember last time we have talked about the adhesions and the focal adhesions जहाँ पे हमने बात किया था, right? So adhesions was like between the two cells. Focal adhesion was between the cell and the ECM, extracellular matrix. Same way, desmosome is between the two cells, and hemidesmosome is between the cell and the ECM. Okay, now let's see how hemidesmosome is formed. So again, intermediate filaments. Let's start from here. Intermediate filaments. Okay, so this is, let me make it. This is like your cell, and ये सारा आपका यहाँ पे ECM. Sorry, ECM. Okay. One second, guys. Yeah. Okay. So let's start making this flow chart of ours. So intermediate filaments are there. Okay. Then we have got the plectin. We'll come to that, like what plectin is. Okay. Now, plectin is again in association with your BP180 and BP230. So plectin in association with BP180 and BP230. Okay. Now, make sure that you understand 230 and 180 में difference क्या है. So plectin is in connections with the here. 230 क्या कर रहा है actually आपका? It is making connections with your intermediate filaments as well as the integrins which is present outside. Okay, so BP230 is making with IF plus integrins which are present outside. Did you get it? BP180 क्या कर रहा है? It is making connections with the plectin and anchoring it to the plasma membrane. Okay, ये इसको हेल्प कर रहा है, या एंकर किया हुआ है. BP180 ने प्लेक्टिन को एंकर किया हुआ है. Okay, where plectin is making connections with the integrins as well as the intermediate filaments with the help of your BP230. Did you get it? And then we have got the integrins B4, alpha 6, integrins present in the extracellular matrix, which is in connections with the other, the components present over there. Guys, did you get it? Should I repeat again? Yes, who would like to try? Akshay, would you like to try?
Yes, sir. So there is interfilaments. So these are in connection with plectins. Intermediate filaments are connection with plectins. So plectin is in association with BP180 and BP230. The BP180 is in connection with plectin, and this help in anchor with the plasma membrane.
Correct.
And next, the BP230, it is in the connection with intermediate filament and the integrins. So integrins are present on the extracellular matrix.
Good, good. Now the question is, what is BP? Okay, so BP was named because these were found mutated in this case in the autoimmune disorder. So BP stands for your bullous pemphigoid. Okay, so what is bullous pemphigoid? It is actually an autoimmune disorder. Okay, in for the skin, जहाँ पे because of this mutation in the skin, ऐसे बहुत बड़े बड़े blisters बन जाते हैं, and they are filled with the ऐसे blisters, like in that inside water will be there, they rupture, then again they form very big blisters, and the entire skin is very, what to say, it's totally damaged actually. Okay, so BP sorry sorry sorry. So BP stands for bullous pemphigoid, and so named because it was first found in this case that it was mutated. Okay, yeah, so that's good. Hemidesmosome is also clear to you guys now. Okay, so desmosomes and hemidesmosomes thus anchor intermediate filaments to the regions of cell to cell as well as cell to substratum contact, similar to the attachment of actin cytoskeleton to the plasma membrane at adherence junctions and the focal adhesions. Remember, we talked about the adherence and the focal adhesions. It is important to note that keratin filaments anchored to both sides of desmosome serve as mechanical link between the two cells in adjacent cells in an epithelial layer, providing mechanical stability to the entire tissue. Okay, so this is same thing, this which we have covered. Okay, the same thing. It's just that little more about the proteins. Now, let's discuss about those proteins which we have mentioned in the earlier slides. So in addition to linking intermediate filaments to the cell junctions, some plaquins link intermediate filaments to the other elements of the cytoskeleton. For example, plectin. So it binds actin filaments and the microtubules in addition to the intermediate filaments. So it can provide bridges between these cytoskeleton components. So later, right now, what we saw was where is that thing? Here. See, right now what we see is that the plectin is only involved in the intermediate filaments and making connections with the integrin molecules, right? Now, but this plectin is also responsible for making connections with the actin filaments as well as the microtubules also, along with the intermediate filaments. Okay, so I'll show in the figure like how this plectin is actually making the bridges between these cytoskeleton components. So all the three are connected with the help of the plectin. Okay, so these bridges to intermediate filaments are thought to brace and stabilize actin filaments and the microtubules, thereby increasing the mechanical stability of the cell. Okay, now two types of intermediate filaments called desmin and the neurofilaments play specialized roles in muscle and nerve cells. ये भी हमने discuss कर लिया. Now desmin, which is present in the muscle, it connects the individual actin myosin assemblies of the muscles both to one another and to the plasma membrane, thereby linking the actions of the individual contractile elements. So again, so when we spoke about the actin and myosin, if you remember, it is a motor protein, myosin, which helps in the contraction, and the sliding filament theory in which we discussed, right, about the myosin role. So there desmin is also present, which is making connections with the intermediate filaments. यानी कि एक cell से दूसरे cell के बीच में जो connection है, and because of which all the cells are contracting together, is because of the intermediate filaments called desmin. Okay. Now, similarly, we have got the neurofilaments present in the neurons. They are present in the long axons of the motor neurons, where they appear to be anchored to the actin filaments and microtubules by the neuronal members of the plakin family. Okay, so what is the takeaway message from this lecture? That we have talked about the intermediate filaments, types of intermediate filaments. So there are 4 types are there. Please go through and try to remember, because these are very important. Every time these type of intermediate filaments will come into picture, Okay, vimentin, desmin, neurofilaments, lamins, nestin, all these are present, and this will be asked again and again. Now let's see in the figure. So you can see that this is actually the figure have taken of the fibroblast and antibody against the plectin. Okay, so it has been colored to plectin जो है आपका वो आपका green color में है. If you see this green color pectins are there. Okay. Then you have got the intermediate filaments blue color, microtubules are the red color. Okay, and then this is the connections where they show like how microtubules and intermediate filaments are connected. Those green yellow things are your plectin. Okay, did you get it? The how plectin is helping in this also, like making the connections with the microtubules as well as the intermediate filaments. Okay. Yeah, that's all. So any doubts, guys, in this question, in this, sorry, not question, any, this topic of intermediate filaments? So among all these three, okay, I'm not saying that this is not important, but please do focus more on the microtubules and actin filaments. Okay,